UChicago's Living Bioelectronics. So picture the bench at UChicago, spring 2024. Not a shiny smartwatch or a rigid PCB, an ultra-soft, skin-like patch that looks almost boring. Inside it, though, is the move that breaks the old frame. Living bacteria, Staphylococcus epidermidis, paired with gentle electronics, acting together as one circuit. Tian's team called it active bio-integrated living electronics, and they meant it. The bacteria aren't passengers. They're the sensing, the actuation, the decision layer, woven right into the device. Here's what it does in plain terms. When the skin flares, the chemistry of inflammation kicks up. This patch doesn't just log the event. The microbes register the local cues like a neighborhood watch with real stakes and shift metabolism that calms the storm. The electronics don't bark orders, they shape the environment and read back what happens. Voltage, ions, metabolites all moving in a loop. The patch is both stethoscope and salve, brain and body in one sheet. And this is where the digital metaphor loses air. There's no central clock telling the system when to compute. It computes because life always computes continuously by flow. Ion channels open and close, membrane potentials drift, tiny voltage gradients ripple across cell layers like weather fronts. Those bioelectrical patterns aren't noise, they're maps. They steer growth, trigger healing, set identity. You don't have ones and zeros. You have graded fields that push cells to decide together what they're going to be. Get this, you can poke the system and it heals. If a wire on a chip breaks, it's done unless you swap the part. Here, the living circuit recruits, rebuilds, and keeps going. Damage is part of the conversation, not a fatal exception. That's because the materials are alive. They sense, adapt, and negotiate. They're not waiting for a programmer. They're doing their own I/O with the world that made them. Let's ground it in the lab. Tian's group didn't just sprinkle microbes for show. They designed an interface that lets the bacteria breathe, feed, and talk, while the electronics stay ultra-soft and skin-friendly. They tuned mechanical properties, water content, and charge so the whole thing sits on tissue like a second layer, not a scab. When inflammation spiked in tests, the community and the circuit shifted states together. The readout changed, the microenvironment changed, and the skin moved back toward baseline. That's closed-loop control without a server farm. Now tie that to bioelectric language. Developmental biologists have been mapping voltage patterns on tissues for years, retina to limb buds, finding that cells use tiny differences in potential to broadcast where and what now. Think of it like a field that encodes a higher-level goal. Make a finger here, close a wound there. The UChicago patch plugs into that layer, not by overwriting it, but by joining it. The control surface isn't an API, it's the body's own field geometry. Why does that beat silicon? Start with context. A silicon sensor samples, it doesn't belong. It needs calibration, sterile separations, rigid packaging. The moment the environment shifts beyond its training set, sweat, stretch, biofouling, it drifts. A living interface leans into the mess. New microbes slide in, the community adjusts, the skin stretches, the matrix flexes, and the bioelectric map rewrites. Noise becomes information. Time becomes part of the computation, not an error term. But wait, here's where it gets interesting. This isn't wetware cosplay. It's a different kind of logic. You don't send discrete commands, you nudge boundary conditions. You set voltages, shape geometry, feed gradients, and let the system find a path. It's analog in the best sense, continuous field-based multiscale. The bacteria metabolize, which changes local pH and ions, which changes voltage, which changes protein gating, which changes tissue state. That chain is the program. And because it's embodied, it's self-documenting. The state of the system is both record and plan. There's no gap between memory and actuator, no file system that can go stale while reality moves. The living circuit is its own ledger. If the light changes or the temperature drifts, the ledger updates with the same physics that will drive the next move. That's why it's robust. It can't not adapt. Let's talk stakes. If you can build circuits that live, you've just erased a line engineers have obeyed for a century. Devices here, organisms there. You've also dodged the brittle edge of the UBC wall. You're not forcing a step-by-step model to capture a fluid, non-algorithmic process. You're partnering with the process itself. The computation isn't simulated. It's happening in the only place it can, inside matter that's busy staying alive. From a design view, this flips the brief. You don't code behaviors, you grow them. You pick host species for specific competencies. S. epidermidis is a skin native. It understands that terrain. You scaffold a habitat that keeps those competencies online. You expose electrodes where the field will speak and listen. You plan for drift, because drift is the feature that finds the solution you didn't know to ask for. And here's the kicker for OSIM. If sovereignty, the system's ability to set and hold its goal, comes from the interplay of morphology, bioelectric fields, and metabolism, then this patch is a tiny sovereign. It senses harm, enacts care, and preserves boundary. It doesn't need an external oracle to tell it what health is. Health is the attractor encoded in the tissue field metabolism loop. That's inception from within, not instruction from above. We're still early. It's a prototype published in Science, sure, but not in every clinic. Yet the direction is clear. The more our devices become part of bodies, literally part, the more they tap into that non-algorithmic layer the wall points to. They stop imitating life and start sharing a boundary with it. And once you accept that, the next question writes itself. If living circuits can heal skin by being skin, what happens when living machines decide how to make more of themselves? Levin Lab's Xenobots, life that builds life. Hold on, because the next move doesn't just blur device and organism. It lets life build life with no genetic memo. At Tufts and the University of Vermont, a small team took cells from the animal pole of Xenopus levis embryos, the ones that would have become skin, and set them free from their tadpole script. No gene edits, no added neurons, just dissociated cells allowed to reaggregate, then sculpted into tiny bodies using microsurgery. They called them Xenobots. On their own, these little clusters started to move. Cilia on their surface beat like oars, and the body planned. The literal shape set how they swam. A plump sphere meandered, a crescent with a mouth-like notch scooped. None of this required a central control loop. The tissue was competent. The cells knew how to stick, push, and coordinate because staying alive bakes those skills in. Here's where it gets interesting. In a dish scattered with loose frog cells, certain Xenobot shapes didn't just bump around. They herded. The C-shaped bodies corralled the free cells into tight piles like a tiny bulldozer circling gravel. After a while, those piles compacted and, without any new instructions, matured into new movers. The parents didn't split. They assembled offspring externally. That's kinematic self-replication, and it had never been seen at this scale in living multicellular systems. Get this, you can change the behavior by changing the body. The team used an evolutionary algorithm to search morphologies in simulation, looking for shapes that would best gather cells. The C-shape popped because the notch stabilizes a vortex of cells like a little cradle. Build that shape in real tissue, and the lab saw the same effect. No one wrote replicate in DNA. Morphology wrote the instruction by shaping contact, flow, and probability. If you've ever coded a robot, you feel the gap. We design planners, controllers, and feedback loops to get a machine to pick up parts and assemble copies. Here, the copies appear because the body's physics and the cell's native competencies align. Agency isn't sitting somewhere in a CPU. It's smeared across the tissue, expressed through cilia, adhesion, and bioelectric crosstalk. The program runs because the parts are alive and the shape makes their work add up. Let's slow down on bioelectricity because it ties this back to sovereignty. Cells talk with voltage. Resting potentials and ion fluxes sketch a map that tells tissues what they are and what to do next. In a Xenobot, that same layer likely coordinates cilia beating and keeps the body coherent under stress. You don't need a brain to hold a goal. The field and the form can carry it. That's OSIM's heartbeat. Morphology as code, field as governor. People asked, is this really replication? It's fair. There's no genome-level template for make a Xenobot. But that's the point. Replication isn't a single trick of DNA. It's a family of processes that produce more of a pattern. Von Neumann sketched kinematic replicators in theory, machines that assemble copies from parts in the world. In biology, we hadn't seen multicellular tissues do it until this. Now we have a case where competence plus shape yields babies by motion. And this shocks AI and robotics for a simple reason. There's no explicit algorithm to port. If you try to digitize it fully, you start losing the edge cases. The way cells stick just enough, how flows gather, how timing emerges from contact. You can simulate pieces, sure, but the living execution draws on capacities that don't silo cleanly. Bioelectric memory, mechanosensing, stochastic search. The irreducible part is the embodied loop that keeps finding a way. But wait, there's a twist in the data. The replication isn't endless. It peters out after a few generations unless you keep the environment primed with parts and keep the morphology tuned. That's a feature, not a bug. It means growth stays under governance by context and form. Sovereignty here isn't unconstrained power. It's the ability to hold identity and produce more of it when conditions permit. That matches how organisms live, bounded by resources, steered by fields. So what did we learn? First, cells freed from their intended body plan don't become mush. They become agents in a new collective, ready to discover tasks their genome never saw. Second, bodies compute with their shape. The same genome in a different morphology yields different goals. Third, distributed intelligence is real. A swarm of cilia, guided by local signals, can write a global outcome: motion, herding, replication. Now tie this to the wall. If you assume reality is a digital tape, you try to capture Xenobots with a finite rule set and a central clock. You'll miss the self-organization as it happened because the system isn't executing steps, it's folding fields, flows, and contacts into a path. OSIM says build with those primitives. Want replication? Don't hardcode, shape. Want control? Nudge voltages and boundaries, then let the tissue negotiate the rest. Here's the kicker. This isn't a one-off stunt. The same lab uses bioelectric cues to grow extra limbs in planaria and to normalize frog faces after perturbation. Patterns remember. Bodies search form space. Xenobots are a clean, undeniable microcosm of that truth. Life can reassemble its own agency when you give it a new body and a playable field. And that means our next machines won't be coded, they'll be cultured. Quantum biology, the hidden layer. So zoom down past the cells we've been talking about, past cilia and membranes into the weird. For years people said quantum effects can't survive in warm, wet biology. Too noisy, too messy. And then photosynthesis refused to play by those rules. In certain plants and bacteria, energy captured by pigments doesn't just wander downhill like a drunk. It takes the best path fast, with efficiency that made physicists squint. Here's the thing. When researchers hit those complexes with ultra-fast laser pulses, the signals looked wavy in time, like a beat, evidence of coherent quantum motion through multiple roots at once. Coherence in a leaf at roomish temperature, not a lab trick in a fridge. The protein scaffold seemed tuned to hold those quantum states just long enough to matter, then hand off the energy with almost no waste. Nature built a quantum router out of meat. Hold on, enzymes do a version of this too. Take reactions that should crawl if they relied on thermal bumps alone. They fly. The best fit is the substrate gets lucky. It's tunneling particles slipping through barriers they shouldn't cross classically. Proton tunneling in hydrogen transfer. Electron tunneling in redox chemistry. The protein flex and local fields line up the shot so the impossible path is the likely one. The rate changes track with quantum models better than with old school ones. And birds. The compass in some species looks like a chemical pair whose spins are linked, radical pairs, sensitive to Earth's magnetic field. Shift the field, the spin dynamics change, the reaction yield changes, the neuron fires different. That's a quantum sensor built into a beak or an eye. You don't need a magnetometer. You need entangled chemistry that decoheres on a schedule biology can read. Why does this matter for OSIM? Because it shreds the clean line between micro weirdness and macro order. The myth said quantum lives down there, sealed off. Biology says no. We'll pull that layer up into mesoscale patterns using structure, fields, and timing. Coherence doesn't need a vacuum if the scaffold filters the noise instead of fighting it. Tunneling isn't random if the protein breath sets the barrier. The control is baked into form. Now tie it to bioelectric networks. Cells sit in voltage landscapes. Ion channels respond to tiny shifts in field, and pH proteins are not dead lumps. They're flexing, polar, capable of changing shape with field cues. So you've got a stack. Quantum events nudged by protein structure, feeding into ionic flows, creating tissue-scale voltage maps that guide growth and healing. It's multi-scale and phase-like. Not binary, not clock, but rhythmic and continuous. Get this, the old digital metaphor wants clean, discrete states. But these systems work by superposition-like sampling, then collapse via structure into the next useful state. That's why life looks nimble. It's not guessing one path at a time. It's sculpted to hold options, then commit fast when context picks a winner. If you try to model that as bits flipping, you drown in state space. If you build with fields and morphology, you ride the tide. Here's a human moment. A plant biophysicist once told me the first time she saw the oscillations in her 2D spectra, she thought the instrument was lying. She reran it, same beat. She changed temperature, solvents, donors. The beat drifted like a living thing. She said it felt like eavesdropping on a conversation we weren't supposed to hear. That's the vibe across this field, structure singing to probability. But wait, this isn't licensed for quantum magic. Scope matters. Coherence times are short, and life leverages them through design, not wishful thinking. The claims that consciousness is a quantum computer leap a few steps we can't take yet. OSIM stays sober. Life uses quantum effects when it can stabilize them with morphology and fields. The payoff is speed, sensitivity, and options under uncertainty, exactly the traits we see in bodies that heal and adapt. So what nobody knew at the time we first framed everything as code is that the hidden engine under some of biology's best tricks isn't binary. It's wave-like, contingent, and context-bound. That engine hands a head start to systems that can sense their own boundary and nudge conditions, because tiny shifts at the right place change the whole flow. That's sovereignty at the smallest scale, control by shaping possibility. Now link back to our pillars. UChicago's living patch speaks voltage and chemistry. Xenobots show morphology writes behavior.